Hello, my name is Zachariah Khan, and today I'll be presenting on bone cartilage interface adhesion energy for osteochondral defects. For some context, the general purpose of the lab is to take a wide range of disorders and diseases to create a sophisticated in vitro microenvironment that can replicate disorders as a platform for trialing therapies. My project will of course follow this guideline. Anyways, here's Timmy, an active 13-year-old boy who enjoys playing tag during recess and his middle school soccer team after school. Timmy sprained his ankle last year, but has ever since complained to his mom of constant pain and locking in his ankle. Getting straight to the point, it's bad for Timmy here. He has the beginning stages of an osteochondral lesion or defect, which is causing some simple pains right now, but will lead to limited range of motion and even further pains as he continues to grow older and continuously is active, as the articular cartilage has no blood supply and it cannot heal itself. All right, so this image here demonstrates an example of an osteochondral lesion. The leftmost image shows the beginning stages where there is loss of adhesion, the beginning of a loss of adhesion. To the right is a more advanced stage where the cartilage has completely broken away from the bone. As I stated, osteochondral defects primarily affect adolescents between the ages of 10 and 20, but is prevalent into adulthood. Additionally, it differs from arthritis, which is simply inflammation within the joints, as osteochondral lesions is the actual breaking away or chipping away of the articular cartilage from the bone caused by stress or trauma over time. Currently, there are only two treatment methods. Corticosteroids or anti-inflammatory medication can provide short-term pain relief, but has no actual healing implications for the lesions. In fact, continuous use of these steroids leads to even worse long-term complications such as osteoporosis and aseptic joint necrosis. The other option, surgery, in the case of someone with a more advanced version of Timmy's lesion, would look like this. This involves surgically removing the lesion and further drilling a hole into the bone through to the marrow as a means of stimulating the stem cells, hoping that those same stem cells will replace the gap the lesion with new chondrocyte cells. A final problem is that attempting to transplant cartilage has complications such as donor matching, as the body's natural immune system often rejects foreign cells. This is where the goals of my research come in. The first goal is to create a synthetic material that mimics the structure of bone. This would eliminate the need for live osteocyte or bone cells. And the second is the creation of a sophisticated microenvironment that can mimic the bone-cartilage interface. And then using that interface, we would evaluate the adhesion energy and mechanical loadings under normal and pathological conditions, as well as the presence of therapies using a blister test. On the left, we can see what the resin substrate will look like, having an inlet at the bottom where a tube will release the air pressure for the blister test. And then on top of the substrate will be a layer of the synthetic bone polymer. Finally, on top of that will be the layer of the chondrocyte-embedded collagen cells, which is represented by the purple, which will be the cartilage layer of the bone-cartilage interface. This is the printer that was used to create the resin substrate. The actual design for the substrate was created by my mentor and partner, Yunte, using a software called Fusion 360. Because this software costs over $1,000 annually, and I don't have money like that, I stuck to Notability as my design contributions. For the creation of the synthetic bone replicant, we had to research biocompatible synthetic polymers and their properties that would be able to mimic the bone. The first polymer is hydroxyapatite, or HA, which is also represented by the formula below. It is a calcium phosphate that composes between 65 and 70% of the structure of human bones. The only issue with it, though, is that when alone, it has low tensile strength and brittleness. This is where our other synthetic polymer comes in. Polyvinyl alcohol, which has high tensile strength and bioprinting capabilities, makes us a suitable partner for HA. For the actual manufacturing of the material, different quantities of HA and PVA were tested. Out of 4 different percentages, the most ideal balance of viscosity to runniness of the material was found with a 15% PVA and 5% HA balance in a 50 milliliter flask. Part of optimizing the material was how long and under what conditions it was mixed on the hot plate, which we found between 65 and 85 degrees at around 75 rotations per minute for about a day. Before we could print, though, we thought of one final alteration for the substrate. Since the only interface we wanted to test was between our synthetic polymer and the cartilage layer, we had to completely fuse the polymer to the substrate while printing. For this, plasma etching, the process of increasing the surface energy of the substrate, was used at a relatively low dose of 75 hertz. Despite being low, this would sufficiently adhere the polymer to the substrate during the next step. As you can see on the left, that's an example of a plasma-treated wall with some paint on it, and you can see there's no chipping, and then on the right, with no treatment, you can see the paint starts to chip away. So that's just a good example of what it would look like. So this is the bioprinter where I printed the polymer on top of the substrate. To the left of the printer is a computer screen where I would input the speed, temperature, and printing pattern desired, as well as commence the printing. A circular printing pattern was necessary for the blister test because when the air pressure is shot through the bottom of the substrate, it will raise up from the center, and any printing lines will not interfere with the strength or quality of the blister. Now that we have our substrate and polymer layer, prep for the cartilage layer could begin. We did a regular collagen and chondrocyte cell culture using a micropipette to deposit the media onto the polymer layer and put it in the incubator, which would help grow and fuse the cartilage layer to the polymer layer to complete the design that I showed at the beginning. I apologize for the white. Now that the design is complete, and it's time to run the blister test, you're probably wondering what a blister test even is. A blister test is a technique for measuring the adhesive energy of polymers, in which the pressure is applied from the bottom of the substrate, pushing, what in the case of my experiment, is the polymer and collagen layers, while measuring the thickness, radius, and critical pressure. We can manipulate the equation at the top to receive the one at the bottom, which will give the adhesion energy. But there's a variable that is missing, which is Young's modulus, represented by the E value, which is the value of elasticity of the polymer. So we needed to run another test on the polymer called a universal tensile machine test. Once all the variables are found, we can plug them into the equation to receive the adhesion energy, represented by G subscript A, of the bone cartilage interface, and compare that value to the normal conditions, which is around 800 joules per meter squared. So the UTM requires a very specific shape, often referred to as a dog bone because of its resemblance to a typical bone. In our case, a type 5 mold is ideal because of its dimensions to best analyze the strength of polymers and plastics when a UTM test is run. After a lot of trial and error trying to create a uniform mold, we found that directly printing the polymer in this shape using the bioprinter was the most effective way to avoid bubbling or any malformations within the mold. So this is what a UTM looks like. There's a screen to the right of the image where the dimensions of the type 5 mold are input and the specific clamp size for the mold is attached. I was able to run one test right before I left the lab, which produced poor results because of the clamp size. So the mold would gradually slip from the machine in small increments, leading to inaccurate data. As seen in the graph, the machine never completed its elongation before it showed a complete trial, meaning that the value of Young's modulus at 879 megapascals is lower than it should be after a complete test. In my discussion, I will touch on what the goal range of the E-value is and my predictions as to where it will end up. Okay, so the previous slides covering the UTM and Young's modulus, that E-value, were all for the purpose of that missing value. Before jumping into the data of the blister test, I want to show you what it actually looks like. Image 1 shows the substrate with the tube where the air pressure will come through. Image 2 shows the polymer and the cartilage cell layer is sitting in the stand, where in image 3, that camera will record those values that in the adhesion energy equation that I mentioned, like thickness, radius, and pressure, all except for that E value, of course. So this is not the actual data of the blister test. The optimization of a crucial value called the PID value, which controls the input and output of air pressure over time on the blister. Looking at the green circle, we can see that the PID value increases the air pressure by 0.5 millibar every 10 seconds until the blister reaches its critical pressure or popping point. The green circle, The green circle has a deposit of 125 microliters of cell media, as seen in graph 1, which as seen in graph 1 leads to an increased critical pressure. The reason we want a higher critical pressure is because it's directly proportional to an increased adhesion energy when plugged into that adhesion energy equation. Looking at the blue circle, we can see that the cell media has been doubled, but the blister critical pressure has been halved. Finally, a similarity between both graphs 1 and 2 is increasing the incubation time that I previously mentioned from 3 to 6 hours, increase the overall critical pressure. So looking at a checklist of the goals that I had at the beginning, for the creation of a synthetic material that mimics the structure of bone, we were able to do that. We were able to create a sophisticated microenvironment that can mimic the bone-cartilage interface. But in terms of evaluating the adhesion energy and mechanical loadings under normal and pathological conditions, as well as the therapies of using a blister test, it's still a work in progress, as we are still optimizing for that Young's modulus value, the E value, and official and critical pressure of the blister test to receive the rest of the values. Like I stated before, we are looking to increase the critical pressure value to indicate a higher adhesion energy. A possible way to do this is to increase the incubation time to 9 hours or even more, but we don't want to incubate it too much, as it will fuse the layers too much, and the cartilage cells will possibly grow into the other layers too much. Additionally, we want to find a way to deposit 500,000 cells, which would be around 1,000 microliters, while increasing the pressure. But as you may recall, the higher the cell count, the lower the blister pressure was. In order to achieve this, perhaps a different step size or PID value is necessary to minimize oscillations so that the air pressure increase is more gradual and consistent. Sorry. In the preliminary UTM test where the E value of 879 megapascals was received, I said that it was too low. This is because the target value is between 2,000 and 7,000 megapascals, which is comparable to the mechanical loading of normal articular cartilage. I was able to follow the linear trend of the graph and predict the polymer will have over 3,000 megapascals for the Young's modulus E value, which would fit in that ideal 2 to 7,000 range. Finally, the reason we want it within that range is because when looking at the articular cartilage, too high a stiffness correlates to impaired shock absorption, and too low stiffness correlates to instability and low load-bearing capabilities. In terms of where I'm going from here with my research, as I have been continuing with the lab as much as possible considering my distance, the first step is to optimize all the conditions so that we receive a normal adhesion value. If this value turns out to be less than expected, this also works in our favor because we can use to observe the effects of possible new drug therapies under pathological conditions, which I believe our lab could do. Big thanks to Dr. Alan Purdy and Yunte for being the best mentors that I could ask for and continuously pushing me. Mr. DeGroat and Mr. Nielsen for putting together this amazing program which gave me this opportunity for my research. My mom and dad for supporting me. My sister for staying with me in DC for a month since I can't rent at 17. And my little brother because he wanted me to shout him out. And the Marine Class of 2024 because it was an amazing experience. Okay, so the question was, what did the PID tuning process looks like? So the thing is, I didn't actually optimize the PID value. That was my mentor because it involved a Python program, and I am not familiar with Python programming. So that was all him, really. Yeah, question in the back. Oh, God. About the cartilage. Okay, so the results of the blister test were less of like actual results where we received those values. It was more trying to optimize that PID value, which would tell us like the correlation between the adhesion energy and how strong the critical pressure was. So we wanted to increase our critical pressure, which is why we didn't actually take any of the measurements from the blister test. So we're still looking to increase that to kind of correlate it to higher adhesion energy, which is what we're looking for. That 800 joules per meter squared value was our target value. Okay, so the question was if this would be like surgically implanted or what the application of this is actually. And so this isn't actually a surgical implant. Essentially what the goal of like a lot of what the lab's work was and what my goal was is we take like these disorders or these problems that are happening in our body and we try and create a more sophisticated version of an in vitro test so that this avoids the need for in vivo trialing. So like we don't need to use animal trials or human trials, and we can replicate this microenvironment, and we can so like the whole thing I was talking about with drug therapy trialing, where we can actually apply like corticosteroids and other forms of drugs and see how that would impact the adhesion energy eventually. That's what I touched on with the pathological conditions when I said that I didn't know if our adhesion energy was going to be lower than that 800 joules per meter squared, because if it does end up being lower, we can test new drug therapies under those pathological conditions, which would be representative of an osteochondral defect, if that answers your question. All right, these next two people are some of the smartest people I know. I'd like to introduce Charlene and Bryn.
Hello, everyone. This is axon degeneration regulation by phagocyte-spheroid interactions.
I'm Charlene Kimon.
And I'm Bryn Mankey.
And we conducted our research at the University of Virginia, Detman and Kusina's labs under our mentor, Sarah Hunter-Chang.
Neurodegeneration, or the loss of neuronal structure or function, is the second leading cause of death worldwide. Think Alzheimer's, Parkinson's, and multiple sclerosis, things that affect a lot of people but still remain quite a mystery to our doctors and our medical professionals.
We thought it would first be best that we give you a brief anatomy lesson of a neuron. First and foremost, a neuron is the reason we are able to process sensory information, to be able to touch, smell, to remember. Neurons make up our nervous system. The axon, the long squiggly part of the neuron, is the path that electrical impulses travel to reach other neurons. And the fragmentation of this axon is a principal characteristic of neurodegeneration. In neurodegenerative diseases, axons die first through this fragmentation process. Axonal spheroids, they look like beads on a string, are hallmarks of fragmentation. The top image is what they look like under a microscope. So I compare them to pimples. A pimple is a break in the skin, and as it inflames, it fills with ick until the skin ruptures. Axonal spheroids are very similar in that they are breaks in the axonal cytoskeleton, and they fill with pathological proteins, calcium, mitochondria, and an unidentified pro-degenerative factor that we will get into later. Now, a signal is expressed on the membranes of dying cells called phosphatidylserine, or PS, because you and I both don't want me to say that a bunch today. PS is an eat-me signal that initiates phagocytosis, or the biological cell cleanup process. Macrophages and Schwann cells are types of phagocytes that are in charge of cleaning up dead cell debris and getting rid of unwanted particles in extracellular space. Spheroids have actually been found to expose PS by Young et al. So, in theory, means that phagocytes could eliminate spheroids.
Sorry, that's bright, and I'm going to warn you, you're going to hear the words Young et al. A lot in this presentation, because Young et al. Is the human who created this three-step characteristic of spheroid formation and membrane permeabilization. So it starts with this latent phase here on the left. You can see that there is an influx of extracellular calcium, so calcium levels inside the axon are increasing, and this is also where axonal transport is interrupted. Then there moves to a transition phase. You can see those little bubbles on the axon. Those are representing our spheroids, and there is a flow of calcium in and out of that membrane. And then once we see these spheroids forming, they begin to permeabilize, which means that that's when the pimple actually is popping or rupturing, and that's when we enter this catastrophic phase. This is the point of no return, when the axon is fragmenting, breaking down, and, which is really interesting, is it's releasing these pro-degenerative factors. This initiates a sort of snowball effect, where neighboring axons are inflamed and begin this process again. At this point, we also see phagocytes, most prominently macrophages and Schwann cells, accumulating around the injury site, but we're not quite sure if they're active in regulating this process or if they're just there.
Now, that was a lot of information in a short period of time, so to sum it up, we want to explore the impacts and mechanisms of spheroid-phagocyte interactions, since this process of degeneration is found across all neurological diseases, and as we said, is still sort of a mystery, which is why understanding how to contain these spheroids could lead to a universal treatment. Our first objective is to determine whether phagocytes can even eat spheroids. Although we know spheroids release PS, so do their surrounding axons. So we don't know whether phagocytes can distinguish between eating spheroids and also their axons. So we will use this PS, the EMI signal, and compare the signal intensities between spheroids and axons, and we do hypothesize that there will be a higher intensity of PS expression in spheroids.
And once we know whether phagocytes are even capable of interacting with spheroids, we want to figure out if macrophages are capable of carrying out this process. So we want to quantify the frequency as well as the degree of overlap in interactions between macrophages and spheroids, and compare the lifetimes of spheroids that interact with macrophages with those that don't, to see if this interaction affects spheroid longevity. Our hypothesis is that since macrophages are more of a secondary responder, kind of in the shadows compared to the primary responder, Schwann cells, that there will be very few interactions and to a very minimal degree between macrophages and spheroids, but these few interactions will significantly prolong spheroid longevity.
And because past data shows that Schwann cells interact with spheroids the most, we want to see if the absence of these Schwann cells will impact the time to nerve fragmentation. To do this, we will compare the neurofragmentation times between Schwann cell ablated or chemically removed Schwann cells in zebrafish versus zebrafish with intact Schwann cells. And we hypothesize that the time to nerve fragmentation will be lower in Schwann cell ablated zebrafish, meaning that the Schwann cells can no longer contain the injury, so the axon will degenerate quicker.
But before we continue with our methods, we do want to note that all handling of live animals was completed in accordance with the UVA animal care policy and with larger guidelines, so only people who were on the correct animal protocols directly impacted these animals. And our mentor, the wonderful Sarah Hunter-Chang, designed and guided our novel experimentation methods, which we performed, and we also quantified and analyzed our data with her guidance throughout the entire process.
To remind y'all, method 1 was about finding out if spheroids can even eat, phagocytes can even eat spheroids. Superior cervical ganglion cultures, or SCG cultures, were established in these things called microfluidic devices, or MFDs, which allow us to grow our own axons. As hinted by the names, these things are pretty tiny, and the grooves that you see in the image there can actually only be seen under a microscope, usually. We were actually given the opportunity to take a road trip and manufacture the molds that make these MFDs. So for our experiment, we made both the molds and the actual MFDs. Basically, the SCGs are plated on one side, and the axons grow across these little ridges to the other. We injured our sample by aspirating them. We basically took a tiny suction and vacuumed the cell bodies out, leaving the intact axons ready to be imaged under a confocal microscope. Now, those images there are what I was looking at during data analysis. The media we fed our samples actually contained dyes. The top image is viewed from the Nexin 5 dye, and the bottom is dextran. The injury was time-lapsed every 5 minutes for 5 hours under confocal microscope. After the images were processed, data analysis was done in the Fiji program with a cross-reference of the two dyes. So Annexin 5 measures the PS signal, and Dextran is for initial spheroid time detection. Weighted averages were used to ensure that the pixel value of each spheroid was normalized, and GraphPad Prism was used to graph our data.
Our second method related to exploring macrophage contact and spheroid lifetime. So we started with zebrafish 3 days post-fertilization on an AB star background. And this is an in vivo experiment made possible really by the translucence of the zebrafish skin. And then we performed a nerve transection, so inducing neurodegeneration, 250 nanometers distal on the posterior lateral line nerve with a micropoint. And this was on the 18th somite. That's just a way of saying where on the nerve this transection was performed. Through our imaging procedures, we captured an image every 5 minutes for 5 hours post-injury so that we could see how this process progressed. And then finally, we did a lot of analysis, starting with a LabKit program to create a 3D surface rendering of the spheroid surfaces and the macrophage surfaces. This allows us to see when the surfaces are in contact with each other and how long the spheroids last, how long their lifetime lasts. Then I built R codes, and I used those codes to quantify the data and produce a few different numbers that tell us about how much these surfaces overlap, how frequently they overlap, and then finally the difference in lifetime between spheroids that experience macrophage contact and those that do not.
Now, method 3 was to determine the effect of Schwann cells during nerve fragmentation. Zebrafish carrying transgenes, which are just artificially introduced genes, were crossed with that glow green, labeling calcium, and red, labeling Schwann cells. Earlier, Bryn went over the dynamics of calcium influx during axon injury, which is why we were able to use a calcium labeling transgene to conduct this experiment. We treated 10 larvae with ranitazole or RDZ for a day, which ablates, chemically removes the Schwann cells, and all larvae, 29 in total, were left in PTU egg water to reduce pigment development for clearer imaging. The zebrafish were then rescreened to make sure the ranitazole did its job, and then the posterior lateral line nerve was transected with a laser, and imaging was done under a confocal microscope. After the images were processed, the data analysis was done blindly, and the time to nerve fragmentation was determined when the last axon of the nerve fragments. There's about 40 to 60 axons in one nerve. Okay, here are our results. The first hypothesis that spheroids would expose PS at a greater intensity than interspersing axons was supported. Looking at the graph, the change in spheroid PS signal over 19 frames, which is represented by the dots, spheroid is in black, and that of axons is in red. As I mentioned before, Annexin 5 labels the PS, and Dextran was used for initial spheroid time detection. And using a statistical test of a 95% confidence interval, we have concluded that phagocytes can selectively interact and eat spheroids.
So now that we know, So now that we know that this interaction is possible, our second hypothesis is that spheroids contact macrophages and have significantly longer lifetimes once they do so. So you can see that there is a very significant difference in the mean lifetime of spheroids that do overlap with macrophages, or at some point in their lifetime touch a macrophage is another way of thinking about it. And that is significantly longer than spheroids that do not ever contact a macrophage. However, there is very minimal overlap with only an average surface area overlap of 3%. So only 3% of the total surface area of the spheroid on average is ever contacting a macrophage. And that's also represented in this graph here where you see that nearly 100% of spheroids experience no overlap whatsoever with macrophages throughout their lifetime. So this is consistent with our hypothesis that macrophages are not primary responders in neurodegeneration, but they could be capable of prolonging longevity of spheroids.
Our third hypothesis that the absence of Schwann cells would impact axon degeneration was not supported. The graph shows the difference in time to nerve fragmentation in minutes between two groups. So the control is the zebrafish with the intact Schwann cells, and of course the ranitazole-treated group has no Schwann cells. Using an unpaired t-test with Welch's correction and an f-test, we calculated a p-value of 0.18, which was not statistically significant. Because Burns data found that macrophages don't interact with spheroids often, but when they do, they pack a punch, we are interpreting that macrophages could be compensating for the absence of Schwann cells, since they now have more space to actually access the injury site.
So there were a few limitations on our research, which again is a novel model, so there are bound to be limitations. And the first is that our samples do have a limited lifespan. After a while, a zebrafish is no longer viable for our research. After a while, it kind of expires. And because of our sample size, our research on phosphatidylserine signal expression is preliminary. Our lab is continuing to investigate this, but what we've presented here is with a smaller sample size. And finally, this calcium influx is something that's relatively unexplored. So the specific dynamics of how calcium is released and absorbed into the axon is not quite understood. But there is a lot of room for further research. First off, we would love to see more repetition with other vertebrates. So far, this research has been done on zebrafish with our model, but we would love to see this research applied to other animals. And then a quantification of phagocytic vesicles, which you can think of as like tiny stomachs in the phagocyte, which will tell you if it has consumed something recently, so we'd know if it's actually like eating up these little bits of spheroid. And that's pre, during, post-injury. That would tell us if phagocytes are eating something when they're near the spheroid. And then the average point in spheroid lifetime where overlap occurs. So maybe macrophages come in at the very last minute, kind of like a final device, or they are preliminary responders who come in right as this process is beginning. We aren't quite sure. And then also whether or not phosphatidylserine is really the necessary signal here to initiate spheroid elimination.
We want to first thank our wonderful mentor, future Dr. Sarah Hunter Chang, who is watching on the live stream right now. Hey. We are forever grateful to have had the chance to experience science alongside her, and we wish her the utmost success in her future adventures. Of course, we thank Mr. de Groot and Mr. Nielsen for putting up with us for these 4 years, for teaching us how to scuba, and for the best marine chem trip ever. We thank Dr. Detman and Dr. Kusinas for inviting us to share their workspaces, and to the members of each lab for their camaraderie and for answering our questions when we could not find Sarah. To our family and friends who sat through all of our practice presentations and supported us across the country as we conducted our research. And finally, to the Marine Chem class of 2024 for their endless support and harsh constructive criticism, and for staying up late with us, definitely not trying to meet deadlines for this class. We are so thankful to have met you all, and we hope our friendships last a lifetime. And here are references. Are there any questions? No questions? Okay, no questions? Okay.
Well, oh yes, you. So the question was about similarities and differences between the zebrafish and the human body. And so my understanding is that the zebrafish genome is very similar to the human genome, but the best benefit of the zebrafish is that with ranitazole, they do have a transparency, and in vivo research like this is difficult to achieve because it would be quite invasive, but since the zebrafish are transparent, we can actually see this process happening through their skin.
Yeah, and the PTU egg water that I mentioned before, we actually let them grow in it, and so that is what reduces the pigment development because it contains this enzyme inhibitor that just blocks the pigment development.
Thank you all very much.
And next up is Betty Roberts proving to us that snails do in fact exist.
I can't hold this, so we're gonna have this like this. Welcome, ladies and gentlemen. I hope you guys aren't incredibly bored yet from those amazing 2 presentations, because I've learned so much from those 2 individuals, and Zach, of course. So today I would like to talk to you guys about assessing post-fire mollusk biodiversity in Opal Creek Wilderness, which is located in Oregon, so just our neighbor down south. Before I get into any of the details, I want to tell you guys why snails should have a special place in every one of your guys' hearts. Snails are vital to our ecosystems because they act as little cleanup crews for our forest floors. Since their diets primarily consist of forest litter, such as dead leaves and logs, they effectively clean up the forest and excrete nutrients such as nitrogen, phosphorus, or potassium, which helps the soil to properly support the growth of existing and new forest and fauna. They are also pollinators and help spread around mushroom spores, but there needs to be some more research on that. So some more about their habitat. Like I said earlier, since their diet is primarily comprises of the forest floor, they're obviously going to be living on the forest floor. And another big reason as to why they are found under logs and leaves is the fact that they need a lot of moisture. So besides being under logs and leaves by some random tree, they like to be next to some riverbeds and other ponds so they can soak up the moisture of their surrounding area. So a little bit of, if you want to do your own snail research and find a snail in your garden and identify it, here's your guide. So snails are commonly identified by a couple of different features. Their first one is their aperture lip, so whether it is flaring out or whether it is just straight down. Another one is how many whorls do they have? So if they have 5, 10, they probably won't have 10. They probably have like 3 or 4. And also the width of their umbilicus, which is on the bottom of their snail, or the bottom of their shell, which is seen in like the topper part. So most of snail research in the past, or malacology, has been primarily composed of these things that I told you, which is very helpful when trying to enter this novel research, since not that many people research about snails. But it becomes more, worrisome since integrative taxonomy is now the standard when it comes to organism research. Integrative taxonomy is just a very convoluted way of saying that we want to know where they are, who are they surrounded by, and what they look like, along with what DNA, like what makes them snails. So to do this, we wanted to go back into Opal Creek and get that DNA and see what these snails actually look like and what their sequences are. So like I said, since we went to Opal Creek, my professor, Dr. Dirks, and Mr. Ansel went to Opal Creek in 2019 with a bunch of classmates and conducted a survey in the Jawbone Flats area. And these were the 3 primary types of snails that they found there. So there was the Vespacola columbiana, Haplotrema vancouverus, and the Encotrema sportella. And although these previous methods of visually identifying these snails, like they did in the past, has been helpful, it's important to go in there again, since you can't see it in these pictures, but Haplotrema and Encotrema, when it gets down to some species, look very, very similar to each other. So going in and taking their DNA is vital to understanding exactly how many snails we have in that region. Just so you guys have a kind of a visual of where we are, that's Oregon, if you didn't know. And in Oregon, there's the Willamette National Park. And within the Willamette National Park is Opal Creek. And within Opal Creek, there's the Jawbone Flats, which I circled for you because you really can't see it from this far away, and because I couldn't find any other better maps. And this is what Opal Creek looked like before the fire, and so around 2019, and this is what Opal Creek looks like right now. There was a Beachy Creek fire in 2020, which absolutely devastated the region and absolutely turned this luscious, beautiful forest into one of rubble and destruction, which, while snails cannot live in that area because of other forest destruction, such as road building and logging, forest fires have a different type of beast because they change the pH level that's there, absolutely destroy the food that they normally consume there, and change the pH level of the soil. I said that already. They change the moisture level of the soil. So my objectives, I had 2 objectives and 2 problem statements and 2 hypotheses. But first we wanted to go back into there and survey how many snails are actually present in the forest now. Although we had hoped for there to be some snails there, we predicted that the species level diversity would actually decrease since the abundance of the snails found there would be gone since there was a fire. And so we also wanted to use a non-invasive DNA collection method because for most of snail research, when trying to collect their DNA, we would cut off their foot or physically extract the snails from the region. And since the snail population there is vulnerable, we wanted to find a way to instead take their DNA and still keep them visually intact and minimize any harm done in the forest. So first things first is we went out back into Opal Creek. My mentors, Dr. Dirks and Mr. Ansel, went back there in October of 2023. I couldn't go out there since it was pretty destroyed by the forest and there was a bunch of falling logs. But they did timed plot surveys, which, like the name suggests, is entailing of creating a plot in the region and for the minimum of about an hour going through the plot looking for the presence or the absence of any mollusks. Then, once they found a snail, they would take a sterilized cotton swab to swab the bottom of their foot for a minimum of 10 times and place that back into an Eppendorf tube. And this is based off of a Mariah paper that we used heavily in our research. After the snail samples were taken back up to Evergreen, the samples were extracted using a salting out procedure. And since it's pretty difficult, I'm going to give you a too long didn't read. So essentially, it uses NaCl to separate the DNA from any debris and contamination. And then we washed out this mixture with ethanol and resuspended the pellet in nucleus freeze water and stored it into the freezer until we were ready to quantify the data. And for most of our research, we used our own buccal cells since we didn't want to go back into the region and disrupt any of the snail cells there. So for most of our time, we've spent doing it on our own buccal cells just to test out the method, especially since, I'll explain it to you guys a little bit later, but since your buccal cells have a lot less DNA than if you took their tissue. So after we got our cells from our cheeks, we nanodropped it and quantified it using nanodropping and PCR amplification. So using the nanodrop, we could just figure out how much DNA we had in the Eppendorf tube and figure out how clean it was. And then using PCR amplification, specifically for the CO1 gene, we could figure out how much, again, we had of it. And also use that to send it to the MC labs for sequencing. And after we received the theoretical sequences, we would manually clean it using the Molecular Evolutionary Genetics Analysis 11 software, or Mega 11. And once they were cleaned, we would align them using cluster W and identify them by blasting them and searching for sequences that are most like the DNA that we uploaded. So, like I said earlier, since for most of our research we use use our own buccal cells, this results that we have right here is based off of physical characteristics yet again. So as you can see, there's a significant decrease in the amount of snails found. We went from about 40 snails found in 2019 to about 6 to 4, and I'll explain the 6 to 4 found in 2023. So some notable differences is that the haplotrema of any type were absolutely not found in 2023, but they were very much present in 2019. And the reason why I said 4 to 6 is because 2 of the anchotrema that were found in the forest were actually dead. So depending on how you want to count it, we only found 1 anchotrema or 3. So this 90% drop supports my first hypothesis that the snail species diversity would decrease because there's fewer snails found. There was only 4 when we got there, which is really concerning since without a healthy multi-species community, few remaining snails face a bunch of different problems such as genetic bottling and breeding depression and demographic stoichicities. So like I said about limitations, since snail research is not that researched, we wanted to keep things on the cheaper side, which meant entailing salting out DNA, which is a lot more difficult But it is cheaper and easier than using a DNA extraction, and I've learned a lot, so I wouldn't change a thing. And also, swabbing the foot of the snail provides a lot less DNA, like I said earlier, than tissue samples. So when trying to sequence the DNA from the swabs that we got of the snails in Opal Creek, it provided a lot less DNA. It would provide a lot less DNA than we initially anticipated based off of the DNA that we extracted from the tissue samples of the snails in 2019. So some further research, or just what I'm doing right now, is analyzing the tissue samples collected in 2019 and comparing them with the samples from 2023. So future studies can also compare the genetic differences. So I'm working with another person in my lab who was introduced in February to use MEGA and sequence and receive the sequencing back from the MC labs. And furthermore, by regularly going back into Opal Creek and just seeing how many snails are in the region as this forestry grows is vital. As you know, I said earlier, snails deserve an important part of your heart. So with that, I'd like to say a big thank you to Dr. Dirks and Mr. Jeff Ansel, since I wouldn't know anything about snails and understand why snails were important until I met them. I'd also like to thank my parents for, in telling me to go into STEM. I wouldn't have been in this program without their support, and also for my older sister for introducing me to Dr. Dirks. I'd also like to thank Mr. DeGroat and Mr. Nielsen for always inspiring me, even when I didn't want to actually go diving because I was scared of the water. They always ensured that I had a great experience, whether that was instead of scuba-ing, I went snorkeling, and I had an amazing time with them. I guess I have to shout out the Marine class of 2024, specifically the RRC, since you guys were pretty cool. And finally, most and not the least, the snails. These are my references. Are there any other questions? In the back. I will be frank, I've never seen Turbo. I actually wanted to do this research since I was really interested in doing the genetic parts of it, so that's what I'm doing right now, and I've had a lot of fun trying to learn MEGA software. I'm sorry, I will watch Turbo when I get home. That's a great question. I would actually not know that. I definitely have to do research on that, but I would assume that you could probably get a little bit of it. But I don't think you would get as much as if you actually swapped the foot. Yes. We would just go back more regularly and hope that there'd be snails in the future, if there weren't any this time around. But yeah, there was a slight. Did you think that there was going to be no snails left, and what were you going to do if there weren't any snails left? And I said that if there were no snails left, we would just go back again and try again. If there are not any more questions, I'd like to introduce the lovely, spectacular, outstanding, I can't keep saying things as you walk slowly, amazing Mr. DeGroat.
Hey, we're going to take a little break, but it's a working break. So what we're going to do is take a picture of everyone who's in the program, students. We're going to meet over on the stairs. Looks like we'll have 3 other extra people, so more the merrier. Starting with seniors on the bottom, juniors, sophomores, and freshmen. I'm going to watch to see how well you organize yourselves and then decide who's going to stay in the program after this. Okay. We'll take that big picture and then a few minutes as a break.
Of the coolest young men in our marine chem class. We have Ben Smoji and Cole Wicklander, everyone.
All right, hello everyone. I'm Ben Smoji,
And I'm Cole Wicklander.
and our project is the design and evaluation of a trap for tracking the settling preferences of adolescent crabs.
All right, so a little bit of background for our project. Crabs undergo a few key stages of development throughout their lives, as pictured in our presentation. They start as eggs and then develop into zoea, which are, I don't know, just like the baby stage. And they float along on just ocean currents until they grow into megalopa, which are a transition phase into juveniles and adults. And also the topic of our presentation. So this transition phase is really important as they go from planktonic to benthic life, which means they stop floating on ocean currents as zoea, and they have to find a place to hide as they molt into juveniles, which are mainly on the ocean floor. If they don't find a place to settle down and hide from predators before they begin to molt and grow, they are easy pickings to, well, you know. So some key reasons for our research. Crabs are a keystone species, which means that they are incredibly important for their environmental health, and they can also be used as an indicator for, well, their environmental health. Crabs are also really important in the state of Washington for their commercial value, as roughly 28.7 million pounds of crabs are are caught every year, and that equates to about $64.6 million, at least in 2023. Some key studies that we looked into for our research was Doyle in 1975 and Zanone et al. In 2016. So Doyle was the one who started everything, and he talked about how larvae are particular when they choose their substrate, as they go from a developmental stage where they grow and develop to a delay stage where they settle and choose to grow into adults. So, and then Zanone et al. Looked at settling preferences of crabs specifically and used a very similar method to what we're going to talk about. However, they focus on flat stones, cobbles, and sand as their three settling as their three substrates, and they did theirs in a controlled environment of a lab, whereas ours was out in the open ocean. The results of Zanone, which we used to formulate our hypothesis, showed that 80% of the megalopa chose either the flat stones or cobble, showing preference for harder substrates. All right, so our problem. There's research about crab behaviors during the zoea stage, relatively little is known about megalopa behavior, specifically their settling preferences, which is weird considering how important this stage is to their life cycle. Knowing this information about the megalopa stage would help, would give us more information about the crab behaviors and give us more effective means of preservation. So our objective in this study, similar to Zanone et al., was to determine the settling preferences of crabs in their megalopa stage of development. And our hypothesis was that they would prefer the bin with the largest rocks due to their greater stability they provide, as shown with Zanone's 80% choosing harder substrates.
All right, for our actual methods. So for choosing a location, we chose Tillow Beach, as it is a marine preserve, meaning its area would be closest to an untouched environment that we could get, and as it is a classic marine chem location. We wanted something that'd be subtidal, meaning it'd remain underwater at all times, and it would be a depth that would avoid potential human interference, while also being easy for us to get at. As you can see, our box location, we were out by some wood pilings that were a handful of yards out offshore. There's a lot of wood pilings in that area, but those were ideal for us. And then for our times that we went out, we were pretty dependent on the tide. We wanted to go out at as low of tide as possible. That way it'd be easiest for us, as this was a pretty labor-intensive project. And we chose 2 periods of time, around 3 weeks each, in order to provide a window for the megalope to settle into their substrate. So for our bins, we selected 3 substrates, one with large rocks, one with cobbles, and one with sands, based on Zenone's study, and based on what a real-world environment would look like. Each bin had a base layer of sand to provide a realistic experience, and the substrates being tested were then laid on top of that. The large rocks, which are 3 to 6 inches in diameter, were collected from Tillow Beach on the day of our trials and placed in the bin, and the cobbles were bought from a local handy store. They were cleaned under running water to remove the dust, and then they were placed in the bins at the beach. And then the sand was all collected from Tillow Beach. When it came to actually placing the traps in the water, this is the layout we decided to go with. So basically our method is we would carry the bins out to the location, and then we'd put them on a kayak, which I was on, while Cole and Mr. Nielsen were pretty much scuba diving. So we'd drag them all the way out to the piling, and then we'd tie a rope onto one of the pylons. We would then remove one of the bins from the kayak and gently place it in the water, slightly opening the lid to allow some water to flow in. That way it would be able to sink. And this would allow the bins to sink to the seafloor easier. From there, Cole and Mr. Nielsen would slowly lower the bins to the seafloor, and once on the seafloor, the lid would be removed and the bin would be connected to the chain, with the cobble bin attached to the pylon and the large rocks attached to the ground with a dog screw. From there, the final step in our methods was to analyze the species that we collected after we removed them. We would take the large bins using a petri dish. We would scoop out 15 total samples, where we would look under microscopes and pick out the little micro species that we could find. Additionally, we would go through the bins manually and collect any macro species. After we went through the samples, we would photograph any species we collected using our phone camera for the macro species, and using a Bluetooth connected microscope to take pictures of the micro species. And we identified macro species as species that were greater than 1 centimeter, and micro species as species that are less than 1 centimeter, roughly. And those are some examples. For results, for collection 1, as shown in the table, the cobble was a more suitable substrate for the micro species that we were aiming to collect. And however, the large rocks seemed to be more preferable for macro species. Arthropods are pretty much your things like your crabs and your shrimp. In our case, we caught a lot more shrimp than crabs, but yeah, and we did not collect anything besides arthropods for the micro species. Echinoderms, those are your starfish, pretty much, which were not an expected grab, but we did get one in collection 2. In general, the trend from collection 1 carried over to collection 2 for micro species. Noticeable difference, though, was that macro species took over in the cobble this time instead of in the large rocks. So overall, our initial hypothesis was that a preference would be shown towards larger rocks, as they are more stable and provide a better shelter. However, in both trials, they were comparable to the results of Zenone's 2016 trial, where a slight preference was shown to stones in general, but a slight preference was shown towards the cobble instead. This is likely because the cobbles are easier to find a settling place in, as there's a much more area and opening to actually get into that substrate layer. And as expected, the sand provides little to no shelter at all, and as such leaves the species open to predators. There was one outlier with the macro arthropod, which was a dock shrimp, but that was also pretty common with their behavior, as they're known to be found in sandy, gravelly areas with swift currents.
Some of our limitations, and specifically our initial trial, We started off in mid-June. We placed the bins in an intertidal location. However, there were strong currents and could easily be interfered with by humans as it was not that far offshore. And because of that, we ended up going back early July to check the bins, and they were gone, which was not what we wanted. And also, because of that interrupted first trial, the second and third trials were delayed to a point that was pretty late in the season that we would be collecting in, and that significantly reduced and affected our results.
Yeah, additionally, it also made us have to spend time redesigning our traps, because our initial layout, we did not have them all connected together. We just kind of placed them. We figured they'd be pretty heavy, because, I mean, these things were, like, heavy. It's not easy to carry one. You weren't going to carry two at the same time, that's for sure. So carrying them out one at a time, they were heavy, and we figured they'd be fine, but the current proved otherwise. And that's kind of another limitation is the labor. These bins were heavy, and they're not easy to carry, especially from the beach to the kayak, and then from the kayak into the water. Not an easy process if you're not trying to tip over the kayak. I was not trying to get wet. Thank you very much. And then some future research. So there's potential with light traps, kind of similar to what Zenone was doing, in a controlled environment where you could catch the megalopes in light traps and then use a controlled environment to test their settling preferences. This would kind of get rid of the real-world experience of it, but it does track what their preferred settlement would be. And then these are our references. And a special thank you to Mr. Nielsen for being our mentor of this project, Mr. DeGroot for helping us along the way, our parents, and the Marine Chem class of 2024. Thank you guys.
And also a special shout out to Katie Vaughn for helping with the analysis of our data and taking pictures when my phone would not connect to the camera.
Yes, thank you, Katie. All right, do we have any questions? All right, cool. Okay, so the question was, would our new design, our current design, how well we think it'd work earlier in the season? We think it'd work pretty well earlier in the season if it was placed out earlier, because we missed like a large portion of the window that crabs spawn in. We're not quite sure if it'd work great because I mean it's still open water. Like the area that we were covering was so little of what crabs could have potentially spawned in that area, so it'd be possible for it to work better, but our method was not very practical, I'd say. Any other questions? Yeah, there are definitely other areas that could be checked out. It's just that Tillo is the easiest to access for us due to our location and everything. There are definitely other areas that could have been accessed, but also it's a great place because it is a marine protected area, and so that allows it to be pretty much away from a lot of human factors for the most part. Julia.
Yeah, so the question was that we attached our bins to a wooden piling to make sure they wouldn't float away. If one being closer to the piling than the other two might have affected the results. I honestly, we don't know. Maybe. Yeah, we didn't look at that very much.
I'd say it's unlikely because, I mean, it's still open water. They weren't that close. It was a fair distance away that it wouldn't have, the pylon wouldn't have directly affected it, I'd say. Any other questions? Cool. Thank you. All right. Next up is our state-bound mermaid, Brandon Lowe. Congrats on being state-bound.
Okay, y'all. Shoot. Oh, no, we're back. Okay. Hello, everybody. My name is Brandon Lowe, and I'm presenting my project, which is evaluating the biodiversity indexes of a marine protected area and a non-protected marine area. Okay, so let's go over what we have so far, or what we really need to understand. So why are MPAs important? So MPA stands for Marine Protected Area, which is defined as a region for long-term protection of marine resources and ecosystems. There are about 127 MPAs in Washington state. And the picture to the right shows the largest ones in the Puget Sound. So organisms in these areas in general are very abundant. So in the article by the Oxford Journal, they debated on if MPAs were effective. Ooh, a little nervous. MPAs were effective and determined that MPAs have an overall increased biodiversity drastically. So one reason for there to have MPAs is to maintain the ecosystems around them. And there's a huge cause and effect of if there is a depletion of these organisms. So if there was nothing surrounding these MPAs, there really wouldn't be much in them. So this pertains to my research because I'm comparing the biodiversity between the MPA and the non-MPA. And the sites that I chose were Titlow Marine Preserve, you know, the one that we all love, and Redondo Beach. So that also goes into why is biodiversity important? Well, it kind of helps us in multiple ways. So it helps us with different materials that we use. It regulates the climate and helps with pollution. It also helps with our water cycle and nutrient cycle. And then it just helps also with anything that we do outdoors. And it also helps with fighting disease and just improving our earth. So this goes into some past data that's already been done. So in 2019, there was a study by some Bellarmine alumni, and they had done a similar project with comparing the biodiversity of Titlow and Redondo Beach. So they found that these two sites were similar with the amount of organisms, but are also different with the amount of human activity they have. So Titlow has a beach that you can walk on, but has no like dock or launching area for boats. While on the other hand, Redondo has frequent human activity through a dock that is a direct launching area for boats. In the past research, they found that Titlow had a higher biodiversity compared to Redondo, and they concluded that this was due to Redondo being an MPA. So after the 2019 research was concluded, COVID-19 happened, and we all know that sucked. And that shut down the Redondo dock, which led to, you know, the decrease of human activity. So this led me to wanting to research more about this topic in 2023 to see if there had been any changes in their biodiversity or not. So this goes into my problem and objective and hypotheses. So the main problem is that there has not been much research since 2019, so this was kind of like a first. And also the Redondo dock was shut down, so it could have been a factor that could have changed the site's biodiversity. For my objective, it was to determine if there has been any change in the biodiversity of Redondo and Titlow with these factors of the closing of the dock and Redondo being an MPA. So my hypotheses are, even though that both sites had different factors like the dock shutting down and one being an MPA, I hypothesized that they most likely would increase in their own sites in biodiversity. I also hypothesized that due to Titlow being an MPA, it most likely will have the higher biodiversity index. This goes into my methods. So, okay, we started number 1, which is, the sites. So when I was determining the sites, I knew that the past data used Redondo and Titlow. So having so it would only be logical to use the past two, the sites that was used in the past research. So we can see that Titlow is on the left. Whoa, no, wrong ones. Titlow is on the right and Redondo is on the left. So I concluded picking Titlow's pilings, which were what Ben and Cole used to conduct my biodiversity. And then I used the east part of the Redondo diving line. So the red lines and the stars are where I would enter the water, and then I would follow the red line out to the sites. And then I did a little rectangle for the pilings. Okay, that goes into number 2, where my path or frame of reference. So first of all, I completed 4 dives in total with 2 dives at each site. And these were conducted in June and September. So I got 2 different seasons of biodiversity counts. I would also use my GoPro to collect my data, and I would use that 20 feet by 20 feet reference, if you could see by the red lines by the pilings. Very nice. All right, this goes into number 3. So after I got my data, I would upload it to my computer, and I would use the NOAA fish guide and Bridget Clarkston's seaweed guide to analyze and determine the species. I would put the number of organisms into a spreadsheet with its species as I counted each one. And then after that, we go into number 4, where after I got my numbers of all the individual species that I had and just individual species and individual organisms, I used Simpson's and Shannon's diversity indexes. So that kind of goes into my results. So I did mention that I used both Simpson and Shannon, so what are they? So basically, Simpson's index is basically a very simple index that you can easily just plug your numbers into. So you just put how many species or the individuals of a single species and then your total population, and then you can get a diversity index. But then for Shannon's, you kind of have to use Simpson's with it, and you kind of do a little bit of calculus, which I didn't really know how to do until this project. I did try to ask my sister for help. Her immediate answer was no. So yeah, that's kind of the basic background of what these indexes kind of are. So this is my data, and the chart that you see above is shows each site and each season and its index. So the arrows show which one is my research and which one's the past research. And in total, Titlow Marine Preserve had 23 different species, and Redondo has 17 different species. You can see that's kind of a big number. As we can see that in the chart, in the summertime, there was a very big difference between the 2 sites, with Titlow being 2 times as much as Redondo. And also, the summer dives showed that Titlow had the highest diversity index compared to Redondo already. So then we go into the fall, and we can see how these indexes individually increased by, like 0.1 to, it's very slim, but they did increase. That's all that really matters. So with that, ultimately, Titlow still had the highest index compared to Redondo in the end, which kind of proved that my hypothesis was supported. So I can also see from the past research that it's drastically increased since 2019, which is really good. So yeah, that's pretty good. All right, this kind of goes into my discussion. So from my research, both my hypotheses were supported with with Titlow still having the highest biodiversity index and both of them increasing from 2019. This was most likely due to the circumstances of Redondo's closed fishing dock, which is what I mentioned earlier, and Titlow being an MPA. So both of these factors definitely increased the biodiversity. Also in my research, a reason for Titlow being higher could have been the amount of species that it had. So like I said, it had 23 compared to Redondo, and because it was like six different species difference. So that could have also influenced my diversity counts. I also concluded that, like I said, the greater number of species that one site holds, the larger diversity index it will have. So, yeah. The effects of these factors show that they definitely have influenced and increased the biodiversity since 2019. So that goes into some of my limitations. So, we all know research, some things happen. So some limitations that I experienced were that it was during the times during the year. So like I said, I conducted these in June and September. They were supposed to be in June and August. So that was a little limitation that I had. And that was because of the fast changing currents. So, Ben and Cole already stated, their bins kind of flew away or, got pulled out and they were kind of gone. So one of the dives that I tried to conduct in August, I did with my dad. And we didn't realize how high the current was, or the fast changing currents were at the time, and we got swept out. And that was not fun. So that would be definitely one of the limitations that I had, because I didn't get any data that day. So then I had to change from August to September. That was also not fun. So that also goes into the visibility of each site. So unfortunately, during the summer, you know how plankton kind of booms because of the sun. So that kind of limited my visibility. Even though I had that 20 feet by 20 feet reference, it still was hard to see. So it was just hard to count my numbers when reviewing the videos. This goes into future research. So I would definitely say if someone wanted to continue this, to do a full year of dives to conduct a bigger image of the biodiversity of each site, if you wanted to use Titlow or Redondo, either one. Also, I would get more than 2 dives, because 2 dives kind of limited the amount of data that I could have collected. So yeah, definitely would say do more than 2. And then lastly, I would get another person to analyze the data with you, because sometimes you can look at one thing and be like, huh, what is that? And then you have to look in the guide and be like, I think that's what it is. So that kind of wraps it up. And I would like to thank Mr. DeGroat for being my dive buddy and going to Titlow when it was super cold, and also like helping me not float away. Mr. and Mrs. Nielsen for driving us around Hawaii. The Marine Chem class of 2024 for their support. Ben, Cole, and Jacob for being the best roomies ever. Thank you, you guys. My family for supporting me with this project and helping me with the dives. And lastly, I would like to thank everybody that got me here today. Here are my references. And yeah, questions. Oh, great. Yes, you. Oh, okay. The question was, how difficult was it to film? So it was kind of difficult at first because of the visibility, I couldn't really see much. But as I got closer to each individual organism, it got better. But yeah, I would definitely say that it was kind of hard to film just because of that visibility. Any other questions? Yes, Bryn. So how did I well, the question was, how did I navigate underwater and how did I basically know where to go? So for Titlow, I basically just kind of swam around the pilings. I didn't really have like a set out like direction or like path. So I kind of just went no in the pilings. And then for Redondo, I just followed the diving line. They have a picture online, which I put into the project, where I did the red arrow. So you just follow that down, and then you come back, and then you get back up to the surface. So it's very easy to navigate. All right. Anybody else? Okay, so the next person is a very important person because, you know, he's kind of related to Mr. Nielsen. And, you know, he's Mr. Worldwide over here, Jacob Hunvin. You're welcome.
Give it a sec. There we go. There we go. All right. Hello, everybody. My name is Jacob Hunvin, and I will be presenting on the effect of changing slope percent on the yield of hydroponically growing romaine lettuce. And I chose to experiment in the field of agriculture because we have a very large issue with not being able to provide enough nutritious food sustainably at an affordable price. And this has caused a lot of issues. According to the United Nations, over 750 million people face daily hunger. And an estimated 1.3 billion people around the world are facing an issue called food insecurity. And that means that they live in a household where they don't know when their next meal is going to be. And a plausible solution to sustainably grow affordable food under harsh conditions is found in an untraditional agricultural field called hydroponics. And hydroponics is different than traditional agriculture because it does not use soil, but instead uses a nutrient-rich water that is filtered through the roots of the plants. And some benefits of hydroponics in comparison to traditional agriculture is that it's very sustainable, it takes up less space, uses less water, and it does not use soil, which are the main issues with traditional agriculture and why they're not really meeting the demand that we need them to. In addition, hydroponics has been found to grow vegetation larger and quicker. So when dealing with hydroponics, there are two main subsystems that are being implemented recreationally and commercially. And that's the vertical growth system, which is shown on the left, and the nutrient film technique, or NFT, showed on the right. I chose to work with the NFT because it's customizable to whatever you're trying to achieve. It's fairly new technology, so further research is still needed to optimize the efficiency of the system. And basically, how an NFT system works is you have this water reservoir that's full of nutrient-rich water, and it's pumped up into the growth tray where gravity brings the water down to the other side of the growth tray and back into the nutrient return. And while that process is happening, the roots of the plants are able to pick up the nutrient-rich water that they need out of the water and use it for their growth. And a specific area in which I want to improve the NFT system is dealing with the angle of the slope tray and how that is going to affect the yield of the plants produced. So some previous research done on this study was on comparing the slopes of different vegetation was by Domica in 2020, and they did a study during the germination period of wheat fodder. And they studied a bunch of different slopes and found that a slope of 6.5% had the greatest yield of wheat fodder. And then another study done in 2023 by Mendonca was a 30-day post-germination period using iceberg lettuce. And they did the same study, but they found that an 8% slope had the greatest yield. So based on this prior research indicating that the optimal slope during the germination period being 6.5% and the post-germination being 8%, potential conflict arises in determining their accuracy. So testing is needed to validate or reconcile these conclusions. And that leads me to my objective and hypothesis. So my objective over the experiment is to test the results of Domica and Mendonca in order to find out what combination of slopes for germination and post-germination provide the greatest yield of hydroponically grown romaine lettuce. And I hypothesized that implementing a system with a 6.5% slope during germination and transitioning to an 8% slope thereafter will produce the greatest yield of romaine lettuce. And that's based off of the studies that have been done before, so we're going to test that out. And just before we get into the controllable variables, just kind of the role of the mentor and me, with the help of Mr. Nilsson, we built 3 identical hydroponic systems, and then after that, it was my responsibility to take them over and monitor them and record all the data that we needed. And the controllable variables that we did is that all this experimentation took place in the Bellarmine greenhouse, which stayed between 24 degrees to 29 degrees Celsius. We used full spectrum LED lighting to signify a July day, as this study was done from November to January. And so we did a 5 a.m. to 9 a.m. We had these LEDs running. And we used a 2-6-in-1 nutrient solution. That's 2% nitrogen, 1% phosphorus, 6% potassium. And all identical treatments were used on all of these, the same systems, like when the water was replaced, just kind of recording data. And then, like previously mentioned, the research was conducted from November 14th, 2023 to January 3rd, 2024. And we used romaine lettuce because it has very good measurable qualities, like the leaf and the height of it and the weight. And it has a really good history pairing well with hydroponics. So as we just continue talking about these variables and talking about the independent variables between each system, we set up 3 systems, each designed to represent the findings based off the previous studies done by Domica and Mendonca. And the table kind of describes the slopes used in each system and the period of life, germination, or post-germination of the lettuce in which the systems were used. So system 1 was representing of Mendonca, who found that 8% had the greatest slope. So during the germination period and the post-germination period, we put the slopes at 8%. And then system 2 was used for Domica and Mendonca, and that represented the 6.5% during the germination that was found from the findings of Domica's study, and then transitioning that to an 8% slope after the germination period. And finally, system 3 was used from Domica, and they found that 6.5% had the greatest slope, so we implemented that to the whole period, germination and post-germination, instead of the germination what they found. And how we kind of ensured the safety of our plants and like giving them the best chance of success is we had some growth maintenance used. We used electrical conductivity to ensure that plants had enough nutrients and also prevented algae breakouts. It's really surprising that an algae breakout can happen in a little water reservoir when I don't even know how the algae got there, but yeah, I don't get it. But we also use dissolved oxygen to prevent plant hypoxia, which is like you don't want your plants to suffocate. And we used pH to ensure and like regulate the acidity of the water. And for the growth measurements of what we actually recorded, we did a weekly collection of the weekly analysis of the number of leaves, and we averaged those out, as well as the total height of all the leaves or total height of all the plants in each system, and averaged those out. And then at the end of the 50-day period, we cut off all of the plants at their right next to their net cup, where they were to the net cup, and we used a dry weight method using a mechanical convection incubator, and we dehydrated all of the plants, and we used an ANOVA test to evaluate our data. An ANOVA test is a statistical test used to analyze the difference between means of more than 2 groups. And just kind of getting into our results, this was during the germination period, what we found out about our germination. I concluded that System 1 had the greatest yield of germination based off the evidence that it had the greatest success of plants germinated out of 20. They had 19. and because it was the quickest system to reach the maximum number of plants germinated. So kind of just concluding that 8% had the greatest yield of plants germinated, followed by the system that started at 6.5% and moved to 8%. But since this is during the germination period, both the red line and the yellow line are only at 6.5%. The yellow line will, after this, be bumped up to the 8%. And then our first part of the results of the post-germination is the average number of leaves collected. And the findings from the data collected over the average number of leaves show that System 1 had the greatest average number of leaves throughout the whole experiment, followed by System 2, and lastly System 3. And this information is important to know as romaine lettuce is a leaf-structured plant, and the size and quality and quantity of the number of leaves is important because that's how romaine lettuce is marketable. And then the second part of our results was the average height of each system. And the data showed that, correlation with all the other stuff, that System 1 had the greatest average height with an average height of 21.8 centimeters. And it's important to note that there's a decrease in the yellow line in System 3. And this is, for whatever reason, one of the plants died during this experimentation. And it could have been to a variety of different reasons, whether that plant was just not getting enough oxygen to the roots or not enough nutrients getting it, but for some reason it died, so that's where the decline in the system is right there. And then dealing with our dry weight, the dry weight data, which basically means we dehydrated all of our plants for 72 hours. I found that System 1 had the greatest average mass, followed by System 2. And as you can see in the graph right there, the blue dots, they represent the mean, and the 2 bars outside of it represent the standard error of the mean, which is used in part with the ANOVA test to determine if the data between the 3 systems was actually different. And I later ran an ANOVA variability test and found that between Systems 1 and System 3, there's a very strong confidence that the groups are different. And we had a p-value of 0.01, which is they're extremely different. We're really confident in that. And then between Systems 1 and System 2, there might be a difference between those 2 systems. We had a p-value of 0.13, which is less confidence, and it's really important that if we got more information on this data, we could have been more conclusive. But between Systems 2 and Systems 3, we had no correlation. We had a 0.57. We could not prove that these 2 were different at all. And basically, what that means is that the data that we collected wasn't just randomly by chance. It actually means that there's a statistical significance between the differentiation between the System 3, which had a slope of 6.5% during the whole duration, and 8% during the whole duration. There is a difference between that. And that kind of leads to the discussion and talking about our hypothesis. These are some photos of some of the plants that we collected. My hypothesis was unsupported. Slope percent of 8% had the greatest yield across all fields of our results: germination, height, leaf count, and weight. And that was followed by the system that I hypothesized, which was System 2, who started at 6.5% slope during germination and then moved up to 8% for the post-germination. And then in last place was the slope that had a 6.5% throughout the whole duration of the romaine lettuce's lifespan. And that brings me to my discussion and kind of with my conclusions. It was really interesting, interesting correlations are emerging as more research is being done on the impact of changing the slope in hydroponics. Most significantly is how the root system of the plants affect which optimal slope would be to affect the growth, the greatest outcome of the growth. And Mendonca, which the results agreed, my data agreed with, used iceberg lettuce, which like romaine lettuce, uses a taproot system. And a taproot system is the image shown on the right. But the data that wasn't supportive with Domica, who found a 6.5% to have the greatest yield. It's interesting to note that they used wheat fodder, which wheat fodder is a fibrous system, as shown on the left. And it just kind of brings up some ideas about how the root system might actually impact which slope would be the greatest for each plant. And some other studies have supported this idea, like Trionita in 2022. They tested the slope of Pokoi plants and found that a slope of 8% also had the greatest yield of Pokoi plants. and those also have a taproot system. So it kind of shows a correlation between that possible idea. And that kind of leads me to my future research. And I think future research, if you wanted to work on this topic, and I'd encourage it because it was fun learning about plants, it'd be to investigate the impact that the root of the plant being grown, how that has an impact on the efficient slope percent. And yeah, and that kind of brings me to my limitations. So the construction of all three systems was obviously not perfect, and we didn't get on the first go. For instance, we used an aquatic sealant to help close off the growth trays to ensure that water wouldn't just run straight out. And there was a lot of leaks at first when we were first creating the system, but it was important that we ran the system before actually putting the seeds in, so that wasn't truly impactful. It just kind of demonstrates that like this system was like a definitely like, while we followed like processes and like previous experiments on this, it was a little bit of a DIY type of thing, as you can see me in my garage right there working on some PVC pipe. And then another limitation to our construction was the placement of the LED lighting. All the plants grew to their closest light source, and we set up 2 LED lights, one on the top and like one in the middle area, and we set them up equally on each 3 systems. But it was just in some situations plants would grow to the downward light because that was closer to it, and that could have some effect on the height of the plant or its root system, possibly connecting to the weight of it as well. And while it is a limitation, it had equal effect on all 3 systems. And then a limitation with the results of the study was when the leaf count and height count were collected. We started collecting the data 10 days after the germination period, so we missed 10 days that could have been recorded. And a correlation of this mistake, like a correction of this mistake, could have been helpful in understanding certain trends, dealing with the transformation from the germination process to the end of the post-germination process. And yeah, and then here are my citations, and then some of my appreciations right here. First off, big shout out to Mr. Nilsson and Mr. DeGroat, really helping us through, and just providing a great experience for our whole class. I got to learn a lot about like scientific method and like conducting research, and it was honestly just a great experience. Miss Savage for opening up some space in the greenhouse and kind of helping me with some resources. Definitely to my family for always helping out and supporting me all of this. Grandma, thank you for always, having great ideas and suggestions. I really appreciate it. And then big shout out to my friends that like would come in and help me like record data. I got Sam over there and Matt. Big shout out to you guys. Thank you for helping me out a little bit because that was took a lot of time to record each and every plant. We had like 60 plants, so it took a long time. And then incredible shout out to the Marine Chem class of 2024. It's been a great experience with all of you guys, and I'm so glad that we've been able to grow together throughout these 4 years. And like, it's this weird thing about like online and Zoom when we met up for the first time, and like, gosh, it was just crazy. So it's great that like I've been able to like make so many memories with you guys. Hawaii was fun. Everything's been fun. Seeing you guys in school, so that's awesome. And then definitely a huge shout out to Bellarmine for supporting this program because it has been a great experience for me and being able to learn and grow from all of this. And I'm glad that we have that opportunity. So, yeah. All right, we got some questions. Yeah, Charlene. Yeah, I saw that from the distaste in my grandparents. I did not get to eat any of the lettuce, sadly. That's something that maybe you can talk to sustainability about, kind of a quick, promo to them. They're growing some in the greenhouse right now using the hydroponic system. Not sure exactly what they're growing, but they're growing something, and it's probably consumable. So go ahead and get it there. But I was not able to eat it because I had to dehydrate the plants for like 72 hours, and they were like dust pretty much. You couldn't do a lot with that. Okay, that might have skewed the data, yeah. Yeah, thanks, Grandpa. Okay, thank you guys.
No, go ahead. All Well, how about one more round of applause for tonight and from last week, too. Nice work. Besides our presentations, one of the products is a research paper. And so they just recently submitted their research papers to the Washington Junior Science and Humanities Symposium. And if anyone gets selected to present at that, that's kind of a big deal. And we've had some people in the past. They also are going to be preparing for the Discovery Regional Science Fair. And that's an online or virtual science fair where, again, they get to present their findings. If the judges like what they hear, possibility of going to the International Science Fair, which is in Washington, D.C. So we'll see how that goes. This is a significant day because this is the last Marine Chem Senior Symposium that Mr. Nielsen will be officially an employee of Bellarmine. He's retiring after, well, he tells me he's retiring. I don't think it's true. I was supposed to retire before him, and I think I still will. But in any case, he's retiring this year, and so wanted to certainly do a shout out to him. He's really the reason I came to Bellarmine. I went to a 20-year party, and they were honoring him. And I saw all this crazy stuff that they were doing and said, I really want to be involved with that. So 3 years later, I came to Bellarmine and came because of this program. And so he's really mentored me and was a tutor to me and helped me grow. And he's always been a really big support. We want to honor him a little bit. His grandson's going to come back up here. He's got a few words. We have a bit of a slideshow. And then I'll bring Ron up, and if he wants to say something, he can. But I'm not going to make you. But I'm going to make it come up here in a few minutes. So you go ahead, and I'll get you a cupcake. And okay, here.
All right, yeah. Not to torture my grandpa, I'm going to, here's some photos first off. It's not quite the full part of it. We'll get to that in a second. But I just wanted to say a little thing that my grandpa started the marine chemistry program in 1986, and he has not aged a day, partially because we don't let him. And as you can see in some of these photos, these are all fairly recent, and you have to think, like, my God, how does he keep doing this? Like, whether we're snorkeling with, like, whale sharks or anything, like, he's always so, like, he's just amazing. And I'm fortunate enough to have him as my mentor on this project, but in reality, he's kind of been a mentor, like, for my whole life. And he's someone that is just kind, loving, hardworking, and always generous. And every day I get to wake up and say that I got the coolest grandparents ever. I'm so grateful for the experiences we've shared together. We can go on for stories, like, for hours. I've been so blessed to have you guys. And I'm also looking forward to all the new memories we will create in our future. Grandpa, you've touched the lives and hearts of so many people. And with all of your care for others, animals, and this planet, you never let me kill any spiders. And it just kind of shows how, like, big your heart is. And Grandpa, you remember when you asked how I got to present last, and I kind of told you that we pick sticks? That was kind of a lie. And basically just want to show you this nice little slideshow that we got for you. Video. So before we hand the mic off to Mr. DeGrow, we'd like Grandpa, Mr. DeGrow, if you guys could come forward, and Talia and Amaris. We got a little something for you guys. Grandpa, yeah, get up. Hearing aids. Come on. Thanks, Grandpa. Love you, Grandpa. We got you guys some nice, I don't want to spoil it. Take your time, don't worry. We got you guys some nice scuba bears as a thank you, yeah, from our class, yeah. And we all signed some of the flippers. Oh, that took a solid 30 minutes to get on, so good luck with that. But yeah, in all seriousness, just from the bottom of our heart, the whole Marine Chem class, I think I speak for all of us, just like, thank you guys so much for all you've done, you know, putting up with us, supporting us through all of our projects. We really appreciate you guys, so thank you.
Thank you, Ron. We love you. I love you. Thank you so much for being a part of our lives. We'll go on, though, but I know you'll still be around. So he'll be mentoring people next year, too. So you folks in the back know that. Well, thank you much, you guys, this evening for coming out. There's still a few cupcakes, if you like. Grab a handful. There's ones with and without peanuts, apparently. And there is tea. I know it's unsweetened, but all the sweeteners to the right. So drive safely, and thanks again. Good night. Thank you. Seniors, we're going to take a picture before you go. One more. This is awesome. This is totally awesome.